some of their work, I'm going to actually let Juliana introduce our seminar speaker today. So Juliana, do you want to take it away?
Okay, can you hear me?
Yep.
All right. Okay, you will not see me because I'm having a little problem with my audio. So, good afternoon, everyone. It's my pleasure to introduce our speaker today, Elena Karlsen-Ayala. So, Elena is a PhD candidate at the University of Florida Plant Pathology Department, and she's co-advised by Romina here at TREC and by Dr. Smith in Gainesville. So she graduated from Smith University. She got her biological sciences degree. And so afterward, she worked a little bit as a research associate in Yale University, and then also as a lab manager at Purdue University. So that was before she started her PhD program here at UF. So Elena is interested in fungi, and so now she's working with pines and mycorrhizing and in fungi and mycorrhizing pines. So I will let Elena take from here.
Okay, awesome. Thank you so much for that introduction. Can you guys hear me okay? Yeah, all right, awesome. So yeah, as Juliana mentioned, today I'm going to be talking about my dissertation research. So from the ground up, the role of mycorrhizal fungi in the pine rocklands. And before I begin, I just want to give a special thanks. I have the privilege of being co-advised by two wonderful mycologists slash plant pathologists, so Dr. Romina Gazis and Dr. Matthew Smith in Gainesville, which means I also have two labs of scientists to collaborate with and work with. And I also wanted to give a special thank you to Dr. Michelle Husino, who's been a really great collaborator and friend and mentor since the beginning. She's a former postdoc in the Smith Lab, and she has now moved on to the USDA Forest Service. So as Juliana mentioned, I really started my love for fungi in my undergraduate degree at Smith College. And part of what really drew me to fungi is just how incredibly diverse these organisms are and how there's really no part of our life that isn't impacted by fungi. So they play many roles as pathogens, as food sources, symbionts, parasites, etc. And that diversity really drew me to this area. And yet, even though this kingdom is incredibly diverse, there's actually a lot of diversity of fungi that we don't know about. So based on our most conservative to our most liberal estimates, there's somewhere between 1.5 and 5 million species of fungi, but we only know about 110,000 described species of fungi. So there's this big discrepancy between our estimated diversity and our known diversity. And so you might be asking, well, so why haven't we described more? We know there are more out there. And that question has multiple answers as to why we haven't really unlocked the full diversity of this kingdom. So for starters, fungi have had kind of a late start to the game. So up until the 1960s, fungi were considered to be part of the plant kingdom. And so it wasn't until the 1960s that fungi were erected as their own kingdom of life. Another factor is that fungi are soft-bodied, which means that they're really not prevalent in the fossil record, which would help us to kind of understand some of the evolutionary history and relationship of major fungal groups. So that's also a lacking aspect. And thirdly, a lot of times when people think about fungi, we think about mushroom-forming fungi or the sexual fruiting bodies of fungi. And while there are many fungi that do form mushrooms, the vast majority of fungi are actually microscopic. And so they do not form mushroom fruiting bodies. And so in order to study and understand these fungi, we have to rely on microscopy as well as sequencing technology. And though we are very fortunate to have sequencing technology at our fingertips, that wasn't always the case. So direct sequencing began in about the 1990s, but in its infancy, it was really expensive and it wasn't available widely. I would say nowadays the sequencing technology has gotten faster and cheaper and more reliable, which has allowed us to really uncover this diversity. And I think something that's really exciting about being a mycologist in this day and age is that the field is definitely moving towards high throughput sequencing, genomic sequencing, and phylogenomics that I think will really start to close the gap on some of that discrepancy between our known and unknown diversity. But something that's really interesting about fungi is that you don't have to go really far to find new things. You don't have to go to remote areas to find new species or even new genera of fungi. There's quite a bit of hidden diversity right here in our backyard. And this is just to give an example of a new genus and species of fungi that was discovered right here at TREC. So back in 2017, a plant sample came into the plant diagnostic clinic with these really unusual truffle-like fungi, which are completely enclosed. So they have lost the ability to actively shoot their spores. And these fungi, when we cut them open, they turned bright red and pink. They smelled really, really bad. And they had these really beautifully ornamented spores. And so this fungus, about 2 weeks ago, the article just came out. We have named this Asparasforus subterraneus. It's a new genus and species of fungi in the Agaricacea, and that has come right here from TREC. And then to highlight some other diversity, Dr. Juliana Soares is working on sampling endophyte species. So these are fungi that live inside of plants. And what she's really interested in finding is whether we can isolate entomopathogenic fungi. So these are fungi that can kill insects that are living in trees as endophytes. And so she has been going around coring trees, some of which are right here at TREC, and looking for Talipocladium, which is a large genus of fungi that are parasites. Sometimes they're parasites of fungi, as seen in this bottom picture, and sometimes they're pathogens or parasites of insects. And so what she's hoping to find is to isolate some new fungi that could potentially be used as biocontrols against insects, or at least to isolate and analyze some metabolites that might be important. And so not to give away too much of her work, but you'll see pictured here some plates of fungal plugs. And it's likely that she will be describing at least one, if not more, new species of Talipocladium from pine trees at TREC. So that's very exciting. So now I'm going to switch gears into talking really about my dissertation research from the pine rocklands. So if we were all in person, I would ask you to raise your hands if you've heard about the pine rocklands, but unfortunately we are on Zoom. But I studied the South Florida pine rockland ecosystem, which for those who may not know, this is a critically endangered ecosystem, which is endemic to South Florida. And what you'll see here is outlined in green is the native range of this ecosystem. And then highlighted in blue is the current range. So you'll notice that this area here is representing the Everglades National Forest, which is the largest continuous pine rockland ecosystem. And outside of the Everglades, there's less than 2% of the original forest intact, and most of the forest is highly fragmented, unfortunately. And for those who might not be familiar with the ecosystem, we do actually have about 8.5 acres of forest right here at TREC. So if you walk along the front gate all the way past the avocados, you'll find the pine rockland forest. I definitely recommend you go check it out. It's pretty cool. And something that I really want to highlight about TREC is that over many years, there's been a lot of efforts to maintain this ecosystem and try to keep it a very functional pine rockland. And it's my understanding that Dr. Schaefer has had a big hand in making sure this fragment has remained as healthy as possible. And that includes prescribed fire, which I'm going to talk a little bit about in a few slides. So as I mentioned before, this ecosystem is critically endangered, and there are a lot of different factors as to why the ecosystem is so endangered. So part of those factors include largely fragmentation and habitat destruction, which is mostly a result of urbanization. Then we also have other factors like encroachment of invasive species, sea level rise, and lack of fire regimes. And all of these aspects coupled together make this ecosystem really important for restoration efforts. So what is the pine rockland? So the pine rockland is characterized by this open canopy of Pinus ellioti variety densa, which is otherwise known as the South Florida slash pine. The understory is a mix of tropical and temperate plants. And aptly named, this ecosystem grows on oolitic limestone substrate, hence the rockland aspect of its name. And as I mentioned before, this is a fire-maintained ecosystem. So for those of you who may not be as familiar with fire ecology and maybe are thinking of wildfires in California, you might be thinking, fire, that can't be good for forests and trees. But actually, fire ecology, as I mentioned, is a critical aspect in maintaining this ecosystem. So as I mentioned before, the pine rockland is characterized by this open canopy of slash pine, which allows some of the more herbaceous, smaller shrubby plants, some of which are only found in the pine rockland habitat in the world, to persist. Now, when you have a lack of fire, what ends up happening is that this ecosystem transitions or there's succession into a hardwood hammock. Now, hardwood hammocks are an important habitat in Florida, but what ends up happening is that this closed canopy habitat ends up being moister and wetter, which makes it harder for fire to come back onto the landscape. And by closing this canopy, some of those trees some of the plants, as I mentioned, that are only found in the rocklands will get kind of outcompeted by these other plants. So fire is really critical to maintaining this open canopy pine rockland. So the South Florida slash pine is the foundational species in this ecosystem. And pine trees associate with ectomycorrhizal fungi, which I will likely be calling ECM from here on out. And ECM fungi are critical to the establishment and the survival of pine trees. However, we really don't know that much about the fungi in the pine rockland forest. They're largely unknown. And for those of you who might be thinking, okay, this is one plant in one forest in one part of Florida. Does it really matter? Should we really care about conserving this tree or even this ecosystem in general? And I do offer the skepticism, and I will counter that with this response and this figure. So timber is worth over $16 billion in Florida. And although I am focusing on the South Florida pine rockland, the slash pine grow across Florida and up into Georgia and Alabama and neighboring states. So the southern variety is the variety that's found in South Florida. You can see its range here in red. And then the northern variety, which is Pinus ellioti variety ellioti, is the northern variety, which is found more in the north. Now, timber, which includes slash pine, loblolly, and several other longleaf and several other pine species, is worth more than any other single agricultural commodity. So that includes citrus, avocado, tomato, strawberry, and blueberry combined, which are some of our largest agricultural economics in the state. So forestry is really important. And yet most of our understanding about these underground interactions between pine trees and their symbionts hasn't been as well characterized as other forests in our country, such as the West Coast. So what is symbiosis? So symbiosis is when two or more organisms are living and growing together. We call this a symbiotic relationship. And this can exist on a spectrum from parasitism all the way to mutualism. So mycorrhizae, which comes from the Greek myco for fungus and rhizo for root, is a mutualistic relationship. And there are two predominant types of symbiotic fungi. There's arbuscular mycorrhizae, or AM fungi, and there's ectomycorrhizal fungi, ECM fungi. And for those of you who might work with crop or other agricultural plants, you may be more familiar with AM fungi, because about 80 to 90% of our plant families associate with AM fungi, which make up about 350 species, AM species of fungi. And these fungi are particularly good at mobilizing phosphorus. But the fungi that I study are ectomycorrhizal, ECM, and about 10% of our plant families associate with ECM fungi. So these include things like pines and oaks. And our estimates are that there are somewhere between 6,000 and 7,000 species of ECM fungi. So a larger diversity of fungi, even though it's a smaller group of plants that associate. And these fungi are particularly good at mobilizing nitrogen. So just to show you what AM fungi look like, they form these arbuscules inside the plant roots. They actually penetrate into the plant root for a direct nutrient exchange between the plant and fungus. And these don't have mushroom-forming fungi, but they do have these beautiful fungal spores that you'll see in the soil. This is an example of some of the diversity of this group. And as I mentioned, a lot of our crop plants like tomato associate with AM fungi. And then there's ECM fungi, which I think are super cute. ECM fungi form these little root tips, which you can see they vary in their shape and size and color. And essentially the fungus is kind of colonizing the outside of the plant root and creating what's called a fungal sheath or a mantle. And this is the area where there's exchange between the plant and fungus. And a lot of mushroom-forming fungi are ectomycorrhizal, although not all mushroom-forming fungi are, but some that you might be familiar with are like Lactarius. Okay, so for plants that associate with mycorrhizal fungi, there are a lot of benefits to the plant, which includes things like higher tolerance for poor soils, drought reduction, protection from heavy metals or pathogen, as well as faster growth and higher nutrition status. And so essentially with this relationship, the plants are providing carbon to the fungi in the form of sugars as a byproduct of photosynthesis. And the fungi in the soil are scavenging nutrients and providing those to plants, as well as helping to regulate water in the plants. So there's a mutualistic exchange going on. And just to show the importance of ECM fungi, particularly in Pinaceae, which is the group I study, we can see that there's been this long co-evolutionary history between ECM fungi and Pinaceae. So here our oldest extant tree family of ECM is about 140 to 180 million years ago. And then our best estimates of the plausible emergence of the ECM lifestyle is highlighted in this purple box. So you can see around the same time that pine evolved, this assumed lifestyle also evolved. And this lifestyle has evolved over 60 times independently through convergent evolution. So it's part of this evolution has led to the proliferation or the diversity of those 6,000 to 7,000 species of fungi. But it's clear that it's a very important relationship that has, survived the test of time. And just to kind of point home about why these fungi are so important, this is just an example of seedlings that are grown with fungi, with their ECM fungi, and without. And you can see without the fungi, the plants are essentially dead. They do not look so good. And so this is, again, illustrating the importance of these fungi. And just to kind of hit home on this point, as I mentioned before, pines are a really important timber crop. And so these pines were introduced into South America as timber production. And when the pines were first introduced, they were introduced without their native fungi. And essentially they failed to establish and didn't really do well at all. But once the pines were introduced with their native fungi, they really took off. And unfortunately, they are so good at getting around now that pines have become a major invasion problem in South America. So I hope that I have convinced you that these fungi are really important. And so with that, I want to kind of go over my overall objectives of my dissertation. So I wanted to characterize the fungal spore bank community on pine seedlings. I wanted to test the fitness of seedlings with native versus commercial inoculum. I wanted to test the impact of salinity on seedling fungal communities and health. And then I also wanted to characterize the mature pine community. So with that, I'm going to start talking about my chapter 1. So for chapter 1, I wanted to assess the ECM spore bank fungi involved in early seedling establishment. So as a pine seedling, you have basically 2 main ways of becoming mycorrhizal as a seedling. Either you can tap into the parent tree's fungal network, otherwise known as the common mycorrhizal network. Or if you establish quite far from your parent pine, you have to establish with propagules in the soil, otherwise known as the spore bank. And so the spore bank is a group of metabolically inactive spores that tend to be long survivors and really good dispersers. And these fungi tend to be the primary inoculum for seedlings. And as I mentioned before, because slash pine is really prevalent across Florida, for this experiment, I really wanted to understand whether the fungal community in the pine rockland was really unique to the pine rockland, or whether the spore banks across these different habitat types were more or less the same. And so for this experiment, I drove across Florida, across these different habitats, sandhill, scrub, and pine rockland. And you can see there are some differences in the plant communities in these different habitat types. And I collected a bunch of soil, and then I air dried that soil for multiple weeks. And this was to basically kill any actively growing fungal mycelium and just isolate the spore bank fungi. And then I took this soil, and using aluminum MySeq, I sequenced the total pool of available fungi in that soil. And then I took that same soil and I used it to plant my seedlings. So I planted both the northern and southern variety of pine on all soil types. So this kind of led to a reciprocal transplant experiment where we could see how the northern variety and the southern variety performed on both northern and southern soil types. So I grew them for 7 months. And then in the end, I harvested the seedlings and sorted them based on morphotype of the root tips. So as I mentioned before, kind of what I mean by morphotypes is that these root tips, you'll see there's a diverse amount of color and shape and size. And part of these characteristics help us to identify these fungi, but I also use Sanger sequencing to confirm the sequence identity. And so now I'm going to go over my results. So over the next few slides, I'm going to show a series of NMDS plots, which is non-metric multidimensional scaling. And essentially what these plots are showing is it's a way to take multivariate data and kind of condense it into 2D ordination space. And what we can glean from these plots are that the closer the points are to each other on the plot, the more similar the fungal communities are, and the further away, the more dissimilar they are. So this first NMDS is taken from the soil data, so the Illumina sequencing of all the soil. And we recovered 1,364 OTUs, or operational taxonomic units. And what we found is that habitat type was significant, and these communities were very unique by habitat type, which was pretty cool to see. This was our first look. Now, when we take the total soil data and we filter out all the fungi that are not ectomycorrhizal, we are left with 188 OTUs. Now, what we can see is still, again, habitat is a significant variable here. But if you'll notice, there's these plots that are also pine rockland, but they're not quite clustering with the other pine rockland soil. So we were kind of wondering what was happening with those plots. And when we take a closer look, we can see that these points actually correspond with our sampling efforts in Big Pine Key, which is a pine rockland site that is in the Keys. So it's not on mainland Florida, but it's in the Keys. And so we're finding that these ECM fungi in the soil are actually clustering separately from the mainland. And so this could be an early look at maybe some island biogeography aspects happening. Now, when we look at ECM from the root tips, so these were the fungi that colonized the root tips of the seedlings, we recovered 22 OTUs. And we also found the same trend that habitat varied significantly. But if you remember, I planted both the northern and southern variety on all seedlings, and we found that tree variety was not a significant variable. So the differences in tree variety did not have a significant effect on which fungi were recovered. And this kind of makes sense because these habitat ranges overlap, and also because we often don't find host specificity at the variety or even sometimes the species level. And then when we look at all the fungi recovered from the root tips, we see that there are some differences between the different habitat types, but that overwhelmingly in all three habitats, Rhizopogon is the most prevalent genus that we found. And this makes a lot of sense. Rhizopogon fungi are generally predominant in the spore bank, and in other systems looking at pine and pine seedlings, they also found that Rhizopogon was the most prevalent. And Rhizopogon are really good at being long-term survivors in the soil. And so with this, to conclude this chapter, we found that the total fungal communities in the soil, as well as the root tips, varied significantly by habitat. We found that Rhizopogon species make up the majority of the spore bank across multiple habitats. And so this work is really our first look at these below ground interactions in this ecosystem, and it was really exciting to see which fungi were around. And so with this, I'm gonna switch gears and talk about my chapter 2, which is my most applied chapter. So this is assessing the effects of commercial inoculum blends on seedling fitness. And so the idea of this chapter was really born out of Connect to Protect Network, which is a really great organization here in South Florida, whose main mission is to basically provide native plants to homeowners to kind of create little stepping stones of native plants between the different patchy fragments. And so there was a survey that was conducted for participants of this program. And the number one thing that the participants were looking for were tips on planting and gardening techniques. And then we also spoke a little bit with some extension agents that are located just around the corner here in Homestead. And they were kind of telling us some stories about homeowners that had really wanted to have native South Florida pine trees in their yard, but they were having trouble with the trees establishing or surviving really well. So they would go to the nurseries and they would buy the South Florida pine tree, but then when they planted them in their yard, they were either the trees were dying or they were very chlorotic, like this one shown in the photo. And so people were looking for more help with how to plant these trees. Now, what the extension agents told us was that they found that for those people living within the native range of the pine rockland forest, they found that some of those trees were doing better than people who were living outside of the native range of the forest. And so essentially the extension agents were recommending that maybe people could buy commercial inoculum blends because some fungi is better than no fungi. And this really got me thinking because, well, not all fungi are equal. So this is just a graph to show these were plants, Pinus eleodii trees that were grown with different scleroderma species, which are ectomycorrhizal fungi. And this group from Chen et al. 2006 collected fungal isolates from multiple different countries. And what they found is that if you pay attention, here is their control seedling. And what's very interesting is that these two scleroderma species, which were collected in China, relative to the control, their total dry weight was actually less with these fungi. And so what this really shows is that A, not all fungi increase the dry weight relative to the control, and also not all fungi are compatible with different plant hosts. So this idea of some fungi is better than no fungi is not necessarily true. And then I also wanted to highlight this meta-analysis, which looked at 28 different restoration efforts using mycorrhizal inoculation. And these, all of the studies in this meta-analysis either had soil from a reference system, so that's like collecting soil from the forest and planting plants on them, or using a single species inoculum, so taking one particular fungal fruiting body and inoculating seedlings with them, or using commercial blend. And overall, what they found is that the commercial blend relative to either single species inoculum or reference ecosystem inoculum was not as productive as the other methods. And also very few studies from this meta-analysis looked at multiple fitness factors of the seedling. So this kind of got me thinking that this would be a really important experiment. So for this experiment, my research questions were, how do the different commercial inoculum blends impact the fitness of pine seedlings? Which ECM species are recovered from the root tips in each treatment? And are pine seedlings able to associate with all of the ECM fungi listed in the blends? And so for this experiment, I bought 5 different commercial inoculum blends, and I collected soil from 3 different forest fragments of native pine Rockland forests. And then I grew these seedlings either on soil, fresh soil from the forest, or on soil that was amended with this inoculum blend. And they grew for 6 months. And then in the end, I took a lot of different fitness measurements, including seedling height, stem diameter, final needle length, root to shoot ratio, percent colonization, as well as root traits like root length and root volume. Now, what I want you to pay close attention to is you'll notice that the boxes that are highlighted in orange are going to represent the seedlings grown on commercial inoculum. The boxes outlined in black are my control seedlings, so they're grown on soil that does not have any fungi. And then in the green is the forest soil, so fresh forest soil. And what you'll notice here is that the commercial inoculum, relative to the control, that looks more or less the same. These seedlings are not as tall, they're pretty chlorotic, they're not as green, and they really look more or less the same as the control. Whereas in comparison, when we look at the forest soil, we see that these seedlings are really green, really tall, really healthy looking in comparison. So now when I show you all of the treatments, I hope that this is a pretty clear trend to see, where all 5 of my commercial inoculums, again, relative to the control, look more or less the same. Whereas my forest soil is, these seedlings are really healthy, really green, and they look pretty good. And this is just an up close so you can kind of see the root to shoot ratio. Again, you can see that the forest soil here, these seedlings are really green. And though you can't see it in this photograph here, these root systems are very mycorrhizal. And then when you look at the control, you have this extensive root system that's not mycorrhizal. And again, the shoots are very chlorotic and not very healthy looking. Now to take a closer look at the root architecture. So these are root scans from the different treatments. And again, you'll see in orange are the commercial blends and in green are the forest soils. And what you'll notice is that the forest soils are incredibly mycorrhizal. So these kind of tree-like dark black areas in the root scan are the root tips that are mycorrhizal. And when you look at the commercial blends, you really don't see much mycorrhization, and the control as well. And so you can see differences in the root architecture. And unfortunately, right now, I do not have the data analyzed from the root scans that would give me all of those root traits. but you'll have to stay tuned for a future presentation to learn more about that. But when we look at percent colonization, so these are counting the total number of colonized root tips divided by the total root tips times 100, we see this trend pretty clearly in that the forest soils are incredibly mycorrhizal, upwards of 80%. Our commercial blend was about 0% colonization, which is great because there are no fungi grown in that soil. And then our best commercial blend had maybe at most 25% colonization. So these commercial inoculums don't seem to be really benefiting the plants. And I wanted to show some of the root tips that we have recovered so far from this experiment. So from the commercial blend, largely what we're finding are rhizopogon species. And you can see there's some diversity in the root morphology of these fungi. And we're also finding Pytholysis. And both of these genera are listed on the blends. But when we look at our forest soil, we can see there's much more diversity of fungal root tips. So we have recovered several different Tomentella, some Cenococcum, also some Rhizopogon, Thalophoraceae, and Sphaerosporella. So there's a lot more diversity, and these data are also pretty new. So hopefully, as I analyze more, we'll find some more diversity on these root tips. But I just wanted to show these because they're really cute, and they really show the diversity of fungi that we're finding. And so really the take-home message from this chapter is that commercial inoculum is likely not appropriate for restoration in this ecosystem. And that kind of makes sense because a lot of these commercial blends are grown in other areas of the country, which would mean that if we were using this approach, we would be possibly introducing non-native fungi into an incredibly fragile ecosystem, which is probably not a good idea, even if the blends were really successful. However, this opens up the possibility for kind of a homegrown blend that could be used using native fungi. So native fungi such as Rhizopogon or Pseuilus could be a viable approach. And just to kind of show, this is a Rhizopogon fungus, and one of these fruiting bodies has millions and millions of spores. And these fungi are really good. They're very reactive and very good at colonizing seedlings. And so you probably would actually not need that many fungal fruiting bodies to inoculate hundreds and hundreds of seedlings for outplanting. And this approach is certainly not a new idea. So just to give you a case study, this is a project with Montana State University in conjunction with the USDA in restoring white bark pine in Montana. So similar ecosystem, not similar to our forest, but similar in that this is the endangered ecosystem. And there was also not much known about the fungi prior to this work. And so their research really attempted to kind of isolate native fungi in the forest and then test different inoculum approaches, either adding, making spore slurries and adding them to seedlings in the greenhouse, or making a soil inoculum. And you can see here that from their inoculum, these seedlings are really well colonized. And so this approach of inoculating seedlings in a greenhouse and then outplanting them in a forest for restoration is really not a wild idea. It's been done successfully in other areas. So I think that this is a great jumping off point for, the future grad student who wants to study pine Rockland forest. Maybe the University of Florida can patent a new blend and we can make lots of money. Okay. So in these last few minutes, I just want to introduce my Chapter 3 research. This experiment is still ongoing, and so I really don't have a lot of data to present to you today, but I kind of want to introduce the background about why we're interested in this chapter. So this chapter is the impact of salinity on ECM communities associated with pine seedlings. So as I mentioned before, even though my research is really largely focused on the pine Rockland system, a lot of the information that will be generated in my dissertation has these broader impacts and implications for other pine systems in the Southeast, which again, we do not have enough data on. And so there are many different coastal pine forests along Florida, as well as along the Gulf Coast. And this just shows different habitats such as longleaf habitat, the South Florida pine Rockland. And this map I really like because it shows you exactly where these pine habitats are. And you can see there are so many habitats that are coastal. And as we know, climate change and sea level, and therefore sea level rise, is a major factor that we're going to have to face in future decades. And what I mean by coastal habitat is that there are some pines that grow right up to the forest, to the sea edge. Now, for those who might not be familiar with pine, pine is not really a very salt tolerant fungi. And so the rise of sea level is going to bring with it salt intrusion, which could potentially have very negative impacts into much of the southeastern forest diversity. And as we know, for those of us who live in South Florida is not a very high elevation habitat. And so even very minor changes in sea level is going to have pretty detrimental effects to the habitat here. And so what we do know is that there's kind of a growing area of research of looking at ectomycorrhizal fungi and salt. So there are some ectomycorrhizal trees, such as Coca-loba, which is a coastal tree that really gets inundated with salt water, that are ectomycorrhizal. And therefore, there must be some fungi that are pretty salt tolerant. And so there's kind of growing evidence that there are some fungi such as Pytholithus and Hebeloma, Pseuilus, Scleroderma that may actually help combat salt stress. And if you remember back to what I said about ectomycorrhizal, they form this fungal sheath, which is kind of like a protective barrier around the plant root. And so there's different ideas of how ECM fungi might be able to mitigate salt stress or salt damage. And one of those might be that some of these fungi are particularly good at sequestering salt in their fungal tissues and preventing uptake by the plants. And there's also other ideas that, you know, ectomycorrhizal plants have higher nutritional status compared to those without, and maybe that will help with combating the stress of salt. But nonetheless, this is a really interesting avenue of how these ECM communities are going to be able to combat salt stress. And so with that, this chapter, I was really interested in, well, how do ECM communities change in response to increasing salt concentrations? Does salinity decrease ECM fungal diversity on seedlings? And are there certain host fungal interactions that persist across the different salt treatments? And what I was finding from my other work in the Pine Rocklands is that even within one forest fragment, there was pretty extensive diversity in the soil type and the soil depth between just, you know, one forest fragment. And so for this, I wanted to design an experiment that was able to test how salt would impact seedlings, but I also wanted to kind of harness the diversity and take a closer look at the finer diversity between trees. And so for this, there are 3 forests that I collected soils, and using 1 meter quadrats, which I placed at the base of 10 different pine trees, I collected soil from 10 different trees in 3 different forests. And then I've subjected those seedlings to 1 of 4 salt levels. So either a control, which is no salt, or 25 micromolars of sea salt, which is approximately 2,800 parts per million, 50 micromolar of sea salt, which is about 5,700 parts per million, and 75 micromolar, which is about 8,600 parts per million. And so, as I mentioned before, this experiment is still going on and there's still a few more months left in it. So I don't have data to show you right now. But what I will say is I'm starting to see an interesting trend between the soils from Big Pine Key, which is that island ecosystem that I talked about in my chapter 1, compared to the other forest soils. And part of what I think is maybe happening with these seedlings is that we measured all of the soils before we planted the seedlings on. We took a lot of soil measurements, which include the EC, the electrical conductance, magnesium, sodium, calcium, as well as pH and some other factors. And what you'll notice is that when I just look at the salts and the EC, the electrical conductance, Big Pine Key, the average of those 10 plots compared to Navy Wells, which is a forest right here in Homestead, and Larry and Penny Thompson, which is a forest in Miami, you'll see that the salt levels, the magnesium, the sodium, and the calcium levels, as well as the EC, are way higher. And so you'll notice that these seedlings are, a little bit smaller. Some of them have died. Some of them have had trouble with germination. And so what I think is happening is that from the get-go, these seedlings are subjected to much saltier soils compared to the other ones. And so while that's not statistically tested at the moment, that's just what I think might be happening. But you'll have to stay tuned for my defense seminar, where I'll have maybe the answer to this question later on. And then lastly, as I mentioned, I want to collect mature pine root tips. So a lot of my other work in my first 3 chapters have looked at seedling dynamics and seedling fungal communities. But what we know from other forest systems is that there's generally a difference between the seedling communities and the mature pine communities. And so I'm hoping that sampling fungi from the mature pine roots will help us to find some more diversity that we may not have been able to see yet in the seedling dynamics, and to help close the gap of our unknown knowledge about this ecosystem, and really provide a great foundation for future research in this ecosystem. And so with that, I want to thank you very much for your time and attention, and I will shift to taking any questions.
Great job, Elena.
Thanks.
Yeah, that was a really excellent seminar. Elena, do you want to handle questions and all from the audience in live time, and I can read some from the chat as they come in?
Yeah, that sounds good.
Great.
I can't even see my chat. Right.
Oh.
I guess I'll start. I have a question.
Uh-oh, a question from my advisor to start.
I'm curious why you think maybe we didn't see more Sewillis. You found lots and lots of rhizopogon, and from some other studies, Sewillis seems to be important as well. And we know there's Sewillis in all the sites. So I was just curious if you thought about why we maybe don't see more.
Yeah, I think that's an interesting question and something I've been thinking a lot about. And I think that one of the kind of hurdles with the pine Rockland ecosystem is that we really don't have a deep soil layer. So I think a lot of people who think of forests think of like this beautiful organic layer and, fungi fruiting. But there are some areas of my ecosystem that like really, there are plants coming from rock. There's maybe a centimeter of soil layer. And so what I think could potentially be happening is that, in order for fungi to fruit, they need to find an opposite mating type in the soil and, have sexual reproduction. And it could just be that the dynamics in the ecosystem make it really difficult for fungi to fruit in certain areas. That's one idea, but I'm hoping that maybe through the mature pine roots, we might be able to recover some of those, because like even the spore bank, which Sewillis is pretty predominant in, I found like 1 or 2 seedlings with Sewillis. So I know they're here. I have pictures of them here, but I have not recovered them yet. So.
Awesome. Thank you.
Yeah.
I'll ask a question. So I'm curious if the mycorrhizal fungal community is ever bad. So it seemed like they weren't the right ones to help the plant grow, but can they have a negative effect on the plant too?
Yeah, definitely. I mean, like I said, these relationships are very intimate, right? The fungi are growing either inside or alongside the plant. And so there's certainly, it's this kind of trade-off between the plant and the fungus to not, go into a parasitic lifestyle. Off the top of my head, I'm struggling to think of an example, but yes, there are instances where they're not beneficial or where that relationship can become more parasitic. And then there's also just fungi that have negative effects on the growth of the plant. Like you pair a fungus and a plant together and the plant really struggles to grow. So yeah, it's not always good is not always good.
Thanks. Romina has a question.
Yeah.
I'll let you go from advisor to advisor.
I know, these were not pre-planned questions. So.
Well, great seminar, super proud. So with what we know now, what we have learned through your research, what would you tell a client, a homeowner that was given a slash pine in one of these events that they give away for reforestation purposes?
Yeah, this is an excellent question because, like I said, chapter 2 kind of shows that the native soil and the native fungi are really important, but in a critically endangered ecosystem, we don't want people just going out and collecting soil to plant their seedlings on. And so I think that this is a really interesting question of how we can maybe sustainably create, blends or inoculum that we can provide people. But yeah, it's hard to tell them, to give them some advice, because I think what other homeowners do is also just fertilize their plants, and fertilizer will actually inhibit these associations. So the mycologist in me would say don't fertilize the plants, but the ecologist in me that's like, we shouldn't just go out and collect soil from an endangered ecosystem, might have to kind of balance those two factors and maybe recommend some nutritional amendments until we can create our own native blend to sell and make lots of money off of.
You
I'll
have
just quickly say a great presentation and great work. And Romina took my question. That's really what I was curious about. I plant a lot of them. So yeah, looking forward to seeing I don't know, some future work.
Great, thank you.
Yeah, I did notice a lot of hands go down after Romina started asking her question.
Well, Braham has one question.
Yep.
Hey, Elena, excellent seminar, very well presented.
Yeah.
I really enjoyed it. My question was kind of related to Romina's. You mentioned, in your results, you see forest soils perform better than in the, commercial mixes. But then you also say there could be a possibility of commercializing what you see. So would you also suffer this? Would your commercial mix suffer the same fate? How would you avoid, or what would the challenges be, and how would you, approach it? Like if you were to commercialize your mix?
Yeah, definitely. So what some of the literature is suggesting is that this kind of trend of the commercial inoculum not performing as well has been seen in other systems. And some more recent research is looking at potentially, double, tripling, going way higher in the recommended amendments for the soil, for the commercial inoculum. So it is possible that at a higher dosage, the commercial inoculum could work. But in my opinion, if it's commercialized at a certain, recommended amount, that should be the recommended amount that works. Now, what I think is happening is that some of these fungi in the blends may not be native to the pine Rockland ecosystem, so there could be some incompatibility there could be some non-spore viability, like if the commercial inoculum have been sitting on the shelf for a long time, maybe those spores aren't active. And so with my proposed approach of having a native blend, if we focused on some of the fungi like Pseuilus or Rhizopogon, which are part of these Pseuilloid group, which are really reactive fungi, those spores can last a really long time. And actually some research has come out to show that Rhizopogon spores actually become more reactive over time. And so hopefully, if we were able to collect some fruiting bodies from the native Rocklands within that fungal group, potentially they would be really good candidates. But that is a good question, and we won't know until we try.
Thanks.
We have time for just one more question, and there is Michelle has her hand up. So
that was an excellent seminar, Elena. You did a great job. I just wanted to follow up on the last question and ask if you have any plans to do any spore viability assays on the commercial blends, or, you know, potentially some sequencing work on those to see what is actually in there, right?
Yeah, definitely. I mean, this is, I have the classic PhD syndrome of running out of time syndrome as I'm projected to graduate in August 2022, and I still have a massive experiment to take down. So the short answer is yes, I would love to do some of these inoculum experiments, but I may have to do, I may have to take on a more facilitator role and find someone who could really lead that project, because like I said, I'm in the running out of time syndrome of my research. But I think that that is a really critical component, and I think that the work from Montana State University really highlights how that work could be done really well in terms of, you know, collecting fruiting bodies and characterizing the different isolates and then testing them both in the greenhouse and then actually outplanting them in the forest and seeing, you know, how these pines persist over time. So I think it would be a great aspect for a PhD student who can kind of pick up from there and take the lead. So. Well, thank you all so much for joining. And if you have any more questions, feel free to email me or find me on Twitter.
thanks for coming. This will be posted online too, in case you missed anything. So
Great, thanks.
See ya.
Bye, Ben. Thanks for coming. My family. Thanks for coming. My fan club from Connecticut. Thanks so much, Dr. Smith, for coordinating this.